Dear student, today we will discuss design of experiment. What are design of experiment? What are their importance? How they can be planned and how you can conduct the experiment in field as well as in laboratory. So it is a very useful branch of statistics by which you can analyze your data which is generated from field experiment or laboratory experiment, and it is a very important branch of statistics and used widely in any research. So let us start. What are the design of experiment and how you can plan a good experiment and how you can get insight from your data using various design of experiment. So first of all, let us introduce what is design of experiment. So scientists in biological field who are involved in research consistently face problems associated with planning, designing and conducting experiments. The basic familiarities and understanding of statistical method that deal with the issue of concern would be helpful in many ways. A researcher who collect data and then look for a statistical technique that would provide valid results will find that there may not be solution to the problem and that the problem could have been avoided first by properly designing the experiment. If you design your experiment properly, then 50% of the work has been done. So let us start. The researcher in biological field or agricultural field or in any field must be carefully planned and design experiments in order to avoid invalid conclusion, and you can avoid the resources which are used in design. They can be wasted if you are not planning your experiment properly. So a poorly designed experiment generally fail to control external factors, and due to which we will get misleading results. So you should keep few things in mind before conducting an experimental design. So these are the key considerations before conducting the experiment. First one is avoid poor design. So what is poor design? If we take an example, suppose you want to compare the mother's milk versus powdered milk, and without controlling for the factor like health of the mother and sanitation, if you are not considering these factors, then you lead biased results. So this is one of the example. In second example, if you want to compare maize varieties without accounting for the say, soil fertility, the direction of sunlight, or the water distribution, if you are not controlling these factors, they can mask true varietal effect from the experiment. Then, in order to do a proper designing, then there are certain steps in conducting a research. There are many phases of the research. So, phases of the research, first one is the concept. You should be clear in your concept. So, concept should be defined properly and it should define the problem properly and their objective. What you want to achieve from this research. This should be very clear in your mind. Second is the design. So, how you are going to control your variations in the field or in the lab. So, you should have a proper plan of controlling the variation in the design which you are using. Third one we will use is data collection. So, Be sure that whatever data is generated from that experiment should be collected accurately. It should be collected accurately. And next important step is analysis. So use a proper statistical method which will answer your objectives, answer your hypotheses. That is very much important in designing or in analysis part. So when data is collected from the field experiment, and it should be properly analyzed through a valid and accurate statistical technique which will answer your questions, and then you disseminate your findings. So dissemination is very much important. Whatever analysis you will get, you interpret the results properly and document these results in a very precise and meaningful manner so that you can convey your findings to the world. So these are the few steps, or these are the few phases of the research. Means you should have a clear concept, you should have a proper design that control the variation, you should accurately collect the data, you should apply proper technique or statistical method to analyze your data, and then after interpreting the results from the analysis, you disseminate whatever findings you will get to the world. So, what are the roles of statistical method? So, statistical technique should guide the entire research from hypothesis formulation to data analysis and their interpretation. So, statistics is the pillar of any research in order to get meaningful insight from your collected data. So, the role of statistical method is very much important. Then another thing you should keep in mind is the controlled conditions. Controlled conditions means your experiment must minimize uncontrolled variation to ensure observed effect are due to treatment, not due to external factor. There are two types of, you can say, variations are available in the data. The variation which a human can control, and the variation which is beyond our control. So our main objective is to minimize the error which is beyond our control in order to get accurate results, or in order to get the effects are due to a particular treatment applied, and these variation is not due to the external factor. So next thing you should keep in mind is formulating questions and hypotheses. These are very much important in designing any experiment. You should be clear in your hypothesis. If you are clear in your hypothesis, and properly defined hypotheses guide experimental procedures which will help you to separate the actual treatment effect from the uncontrolled variation. If your questions are not clear, or if you are not properly set the hypothesis, then it is very difficult for a researcher to get valid results. And then, of course, in conclusion, you can say good experimental design combines technical knowledge, statistical expertise, and careful planning in order to ensure valid, reliable, and cost effective results. So, these are the few points you should keep in mind before conducting the experiment. Means, first of all, you should have a proper research question. You should be clear in your mind that what I want to do. Then, on the basis of that research question, formulate proper hypothesis. Then third step is conduct the experiment in accordance with a proper statistical technique so that you can get valid and cost effective results. So, there are certain features of experimentation. So, let us start with these. The salient features of experimentation. Number one is control of extraneous variation. So I already explained that there are two types of variation in the data. One variation is due to the treatment you are applying, and other variation is the variation which is beyond our control. So all the variation in the data except those due to treatment under study should be controlled or eliminated. Means extraneous variation should be either controlled or you can eliminate by the means of certain techniques. And always avoid artificially created condition that favor one treatment over other. So by doing these things you can control the variation which is beyond our control. Then number 2, choosing the right design. The design of an experiment significantly impacts the ability to detect treatment differences, right? So selecting a appropriate design before starting the experiment is very much important in order to get meaningful results, right? Number 3, adequate replications. The number of replication, how many times your experiment is repeated under identical conditions is very much important. If you use a particular method, then you have to ensure sufficient number of replication for each treatment must be there in order to get valid inferences, because without replication in experimental design it is not possible for us to get the error variance, which is the key component of any experimental design. And fourth one is random allocation of treatment. Means you should apply the treatment at random to the various experimental plot without favoring any treatment or without disfavoring any treatment. So treatment must be randomly assigned to experimental unit to avoid, you can say, you should not be biased towards a particular treatment. And if you are biased, then you can't get the valid results. So unbiased method of allocating treatment to the experimental unit is very much important. Then let us define what is the experimental design. So the design of experiment is a sequence of steps which I have already explained, taken before the experiment to ensure number one, appropriate data collection, means you can appropriately collect the data. Then objective analysis, what kind of research question you are asking, whether your design is capable of answering these questions. Then number 3, the valid inferences regarding the research problem and what inferences you can draw from the data which you have analyzed through a proper experimental design. And number 6 is understanding objectives. The individual formulating the design must have clear understanding of the objective which you want to achieve in order to ensure the experiment addresses the research question effectively. So, objective is very much important. Of course, selecting a design is very much important. Then, random allocation of treatment is very much important in order to reduce the bias. And you must have adequate number of replication in order to get the valid results, or you can get the precision of the results properly. Then what are the basic principle of design of experiment? In any experiment you should follow two or three principles which are very much important in order to get valid results. So what are these basic principles of experimental design? So Professor Ronald A. Fisher outlined three fundamental principles in order to ensure the validity, accuracy, and interpretability of experiment results. Means if you follow these three basic principles, then you will certainly get valid result, you will certainly get accurate result, and you will properly interpret your experimental results. So first principle of experimental design is the replication. So what is replication? Replication means repeating treatment across multiple experimental unit. Means a single treatment is repeated a number of times in experimental unit. So replication is nothing but repeating treatment across multiple experimental unit. Number two is the randomization. Randomization means assigning treatment to the experimental unit by chance, not by your will. Means every treatment must have equal probability of getting a particular experimental plot or experimental unit. You should not be biased towards the treatment. So randomization is a process by which you can assign these treatments to the experimental unit by chance. So randomization means assigning treatment to experimental unit by chance. Then third component is very much important, which is local control. Local control means you adopt certain practices by which you can make your experimental material homogeneous. So you should divide sometime in field experiment it is very difficult for us to maintain the homogeneity. So in order to maintain the homogeneity, we apply certain local management by say dividing experimental material into homogeneous blocks to reduce the variability. So, if you take an example, you can find out in an agriculture experiment, the blocks or the replication may be formed based on the soil fertility gradient. So, in soil or in field experiment, we come across the situation when the fertility gradient is not uniform among all the experimental unit, and it is in one direction the fertility gradient is more, in other direction the fertility gradient is less. So we will make block in such a way that every treatment should have equal chance of getting any plot which have more fertility gradient or which have say less fertility gradient. So let us explain these three principle in detail. So first principle is our replication. So what are the replication? Replication means repeating a treatment multiple times in an experiment. And suppose if you want to repeat a treatment say R times, the design is said to be have R replications. And why these replications are important? There may be a question why these replications. There are so many reasons uh, why using replication in experimental designs. They are number one, it increases the accuracy of the experiment effect estimates. In order to estimate the effect of a treatment, and if that treatment is replicated say R times, then of course by single observation and by repeating the experiment R times, the estimate obtained from the R unit is more reliable as compared to a single one. Then it provides an estimate of error variance. It is very important. In an experiment, if you are not capable of estimating error variance, then you cannot draw meaningful inferences, which measures the differences among observation under the same treatment. So, increasing replication too much is not practical. Now you can ask, we will repeat the experiment say 20 times. It is not practical because if you increase the number of replication, it also increase the cost of experimentation. and it requires more resources. So the number of replication should be optimum one, not much large, not much small. So these replication, number of replications should be decided on the basis of number one budget and available resources, which is of course very important. Then number two, required precision, what precision you want to estimate, and what level of precision you want to estimate various effects or results. Then sensitivity of the statistical method like t-test or f-test, because the t-test and f-tests both are depend upon the number of observation or the number of degrees of freedom. So if number of treatment is less, the error degrees of freedom are obviously less. That's why these two tests, which are generally used in experimental designs, they are very sensitive to less number of degrees of freedom. Okay. Then replication ensure reliable results with balancing, sorry, keeping the cost and precision. You should make a balance between the cost and precision. Then how you can determine the number of replications? This question may be asked. Of course, when replication are so much important, then how will you will decide which number, how many number of replication we should keep for a experiment in order to get reliable results as well as cost is minimized. So the number of replication in an experiment always depend upon the error variance. Okay, and the desired level of precision, at what level of precision you are want to estimate the results. So if lower error variance means higher precision, if you have less error variance, then of course you have higher precision. So replication is used to detect meaningful difference between the treatment. So they can help us to detect meaningful differences between the treatment. So if a researcher want to identify whether two treatments differ by a certain amount, say D, the number of replication you are using in that experiment is R, can be calculated by this formula. So this is actually T statistics T0. This is the difference which you want to a certain amount, and this S is the error variance, R is the replication. So from this formula, you can find the R, which is nothing. If I square both sides, then I will get, this is T square will be equal to D square upon 2S square upon R. So if I solve this in terms of R, then I will get R is equal to, say, T square. This D square will goes here, so it is D square, and then multiply it by 2S square. So you can determine R if you have a certain level of significance, and if you want to compare the certain amount of difference, say D, and S is the error variance. So by this formula, you can identify the number of optimum number of replications, where T0 is the critical value of T distribution, and it is oftenly taken from the table at a certain level of significance. S square is the error variance, and D is the minimum detectable difference between treatments. Okay, so with the help of this formula, you can ensure that difference is greater than a particular value D are statistically significant. Or if S square is based on large number of observation, T can be approximated as normal variate. And for smaller sample sizes, T depend upon the degrees of freedom. So by this approach, researcher can decide the number of replications needed to achieve a desired sensitivity and precision in their experiment. Right? So let us take an example by which you can estimate the number of replication for a particular level of, say, for a desired level of precision. So an experiment on wheat crop was conducted to study effect of application of nitrogen and phosphorus on yield in randomized block design with three replications. This is my problem statement. And there were 11 treatments, two of which are, say, 60 kg per hectare of nitrogen and 120 kg per hectare of nitrogen. And the average yield figure for these two application of fertilizers were, say, this and this. When you apply 60 kg of nitrogen, you will get this yield. When you apply 120 kg, you will get this. So, and it is required that differences of order of 150 means if difference between these two is greater or equal to 150, then we should say it as significant difference. So, first of all, the error mean squares S square was supposed 1213.88 EMS, assuming that experimental error will be the same order in future experiments and your critical value of t is 2, the error of degree of freedom is 30 and there are 11 treatments as I have already mentioned. Clear? And I am interested in only two treatment where this amount of nitrogen was applied. So, how many replication you can get from this experiment? So, you can get by R 2 T0 square S square upon D square. 2 T0 is your 2, so it is 2 square, and this is your S square, error mean squares, and 150 is the desired difference and its square. So, you will get 4 replication approximately. Thus, an experiment with 4 replication is likely to bring out the differences of order of 150 kg per hectare as significant. So, this is the way by which you can decide the number of replication. Another criteria for determine of R is to take the number of replication which ensure at least 10 degrees of freedom for the estimate of error variance in the analysis of variance. Clear? Of the design concern, since the sensitivity of the experiment will be very much low as F test, which is used to draw inferences in such experiment, is very much unstable below 10 degrees of freedom. So your F test is less stable at for the degrees of freedom which are less than 10. So in order to get at least 10 degrees of freedom for error, the R can be decided. Then second part, which is randomization. So what is randomization? I have briefly defined the randomization in introductory part. So randomization is the process of assigning treatments to experimental unit by chance. Do if we apply the randomization process, this process ensures that every treatment has an equal chance to show its effect. Means there is no favorism, even when we don't know the variability of the experimental material. So, in any experimental material, if we assign the treatment randomly, means by chance, means any treatment can be occupy any plot, then this process is known as randomization. Why this randomization is important? Number one, for valid statistical test. It is very much important. Means randomization ensures that statistical test, which I have already explained, like t test to compare two means, f test to compare multiple means, should be valid. Okay, these tests depend upon the assumptions that data follow a statistical distribution, and randomization helps us to meet these requirements. Okay, so randomization is very much important for obtaining valid statistical test. Number two, it is also important for avoiding bias. So randomization process protect the experiments from unexpected factors like changes in temperature, soil fertility, or equipment error. So it ensures that these uncontrolled factors operates randomly and do not favor any particular treatment. So due to which you can avoid the biasness in your experiment. Then you should keep this thing in mind that randomization alone is not enough. It must be combined with replication, means repeating treatment multiple times to allow the use of statistical tests like t test, f test in order to get valid conclusion. Right? So we have studied the replication, means repeating the experiment in order to get the valid results, or the precision of the result should be increased, and randomization is a process by which you can reduce the bias. And third component, which is of course very important in field experiment, is the local control. Local means local, controls means controlling, means you apply certain techniques locally in order to manage the heterogeneity in the field. So in agricultural experiment, if field is not uniform, means it is heterogeneous, not homogeneous, so random allocation of treatment across the entire field can lead to increased experimental error, which is due to soil differences, temperature differences, and so on and so forth. So, in order to reduce this error without increasing the number of replications or without disturbing the randomness, local control is used. Means we apply certain local techniques in order to reduce such kind of variability. So, this is the local control. So, how local control works? How local control works means the experimental field first divided into homogeneous group. Then question arises how you can divide a heterogeneous field into homogeneous groups. So that can be divided with the help of the fertility gradient. If you study the fertility gradient in the soil and you find that the fertility gradient is in a particular direction, so you make the replication perpendicular to the fertility gradient. So the fertility gradient is divided into various blocks. Now in each block we randomize the treatment, so every treatment has equal chance of getting a plot which has high fertility gradient and which has a low fertility gradient. So by this you can apply the treatment. So the experimental field is divided into smaller homogeneous group or blocks or replication, based on the soil fertility or other factor which is not uniform in your field. And these blocks can be arranged row wise, column wise, or both. And row wise, column wise, and both, they depend on the fertility gradient of the field. Suppose if your fertility gradient is in, say, row wise, then you make the plot column wise. If it is column wise, then make you plot row wise. If fertility gradient is not known to us or it is diagonal effect, then you can use both row and column as blocks, right? So treatment are then randomly assigned within each block, and this method ensures variation within blocks should be minimized while variation between blocks are maximized. So if you make these arrangement, the effect of soil fertility differences are separated from the experimental error, which makes us to detect smaller differences between the treatments. So let us take an example of experimental design using local control. First one is randomized block design. This is a kind of design we will discuss later on. So it eliminate variation in one direction, means row wise. Number two, Latin square design. Latin square design eliminate variation in two directions, row wise and column wise. And local control is an essential technique or tool to improve the accuracy of the experiment conducted at heterogeneous field or in field condition. So it is very easy as to conduct an experiment in laboratory where the condition can be homogeneous. So now we will move towards, after studying what is experimental design, what are its key component, it may be your replication, it may be randomization and local control. Now, how you will plan a good experiment. So, what consideration you should keep in mind for planning an experiment. So, planning consideration for field experiment. To ensure accurate and meaningful results, the following steps should be considered when planning field experiments. Means there are n number of steps you should follow. These steps, if you follow these steps properly, then your results are very accurate and you can draw meaningful conclusion from these results. First one is, I have already mentioned, objectives of the experiment. Means clearly define and you make the priorities of your objective to design a focused and realistic experiment, right? So you should define your objective according to their priorities, and design should be well focused and it should be real also. Number 2, the scope of the experiment. What does it mean? So you should keep two things in mind in order to this point. Mean you should have understand the agricultural, and you should have the statistical aspect of the problem in order to ensure the experiment is relevant and comprehensive. Means I mean to say you should have a good agricultural background if you are conducting a agricultural experiment. On the same time, you should have a sufficient amount of statistical knowledge in order to achieve your objective. Because if you choose a wrong design, if you choose a wrong technique, then it is very much difficult for a researcher to achieve his or her research objectives. Number three, you should clearly define your hypothesis. Means formulation of hypothesis is very much important. So you develop hypotheses based on your research objectives, and these hypotheses should be very concise and clear. So your research objective which can be tested, the hypothesis may be accepted or may be rejected depending upon the results. Number 4, the selection of the treatment is also very much important. So you choose treatment that address your objectives, such as comparing new selection with standard variety in a breeding program, or comparing new drug with the existing one for a particular treatment, and so on and so forth. Then fourth one is selection of site and experimental material, the place where you want to conduct your experiment, and what are experimental materials is available with you. So the site should represent the target population, and material should be homogeneous and relevant to your objective. So there are few more. These are: number 6, selection of plot size. What size of plot you should select? What should be its shape? Whether it is a square, whether it is a rectangle, whether it is maybe of any other shape. And block. So conduct uniformity trials to determine the optimum plot size, its shape, and divide your whole field into blocks to minimize the soil heterogeneity, right? Then decide about the number of replications. The number of replication is, as I have already mentioned, is very much important. So decide the number of replication based on your precision, what precision you are requiring in your experiment, what is the field variability, what are the resources you have, and what is your plot size. So ensure at least 10 to 12 degrees of freedom for error in order to get valid results. Then eighth is choice of experimental design. It is very much important. So choose the simplest design that meet the requirement, precision, and your resource limit. So decide whether the design is unifactor or factorial, and whether it eliminate one or two level of heterogeneity. So depending upon your field condition, depending upon how many factors you want to study in an experiment, it should be unifactor, means a single factor experiment. It may be multi-factor experiment, which comes under the category of factorial experiment. Or if your field has, so say, fertility gradient in unknown direction, then you want to remove two-way heterogeneity. Under such condition, we should use Latin square design. We will discuss all these things when these designs are studied. Then next one is transformation. Means it is not necessary that you will get normal data from the field experiment. And assumption of normality is very much important in analysis of variance. If the assumptions of ANOVA is not met, so in order to make the data normal, we will use certain transformation. So you should use, or you should transform your data to a suitable scale for valid analysis. So there are so many transformation that we'll also discuss in subsequent sections of this course, that which transformation apply which transformation at a particular condition. Then statistical analysis. It is very much important. Analyze the data using ANOVA and additional method which answer your research problem. And 11th is drawing conclusions. So after analyzing the data, you should have certain capability to interpret the results of that ANOVA. and present them in a clear report. So these are the few consideration before conducting the experiment. I am again emphasizing that the statistics must be consulted before conducting the experiment. Nowadays, the experimenter conduct the experiment, then they go to the statistician and ask them a suitable design, what I should use so that my research question can be answered. This is a wrong practice. You should consult the statistician before conducting the experiment. So these are the few planning consideration good for a good experiment. Then, of course, when we study this course, then statistical terminologies are used in this course. What are these terminology? You should be aware of these. First one is experiment. An experiment is method to find answer of a specific problem. And a experiment can be absolute experiment which determine absolute values. For example, average IQ of a group or correlation between two variables. Then comparative experiment means you want to compare the effect of two or more factors. For example, different fertilizers, crop variety, or medicines. Then second term is treatment. So treatment is a controllable factor applied at specific level by the experiment, and the treatment may be fertilizer or pesticide for crop if you conducting a agronomic experiment. It may be drug concentration for animal science experiment. It may be irrigation levels for agriculture engineer and crop varieties for plant breeder. Then next is experimental unit. It is the smallest division of experimental material that receives a treatment. For example, if you conduct a study on a drug effect on various rats, so rat is your experimental unit. In field, the plot which receives a particular treatment is the experimental unit. So, plot of land or batch of seeds, agronomist consider these things as experimental unit. Group of animals, animal scientist can consider group of animal as experimental unit. Tree species in forestry research, so tree may be considered as experimental unit. Then experimental errors. So these are the differences in results. For example, if you want to study the yield from experimental unit treated the same way are called experimental. These error can be occurred in an experiment due to various reasons, and these are number one, variation in the material, number two, measurement or observation error, or number three, the external factor not controlled in the experiment. So we try to reduce these experimental errors. So how you can reduce these experimental error? So you should use homogeneous material, properly stratified, careful execution, and efficient experimental planning can reduce these experimental unit. Then yield. Here yield is not the yield of a crop, it is measured outcome. It may be crop yield from different experimental unit. The number of tillers is your outcome variable from an experiment. The number of tiller is your yield or the number of tiller you can use. Replications, again repeating an experiment or treatment multiple times to ensure accuracy and reliability of the results. Then another important definition is precision. So precision refers to the amount of information in a design, and it is reciprocal of the variance of the mean. So, in an experiment, if it is repeated r times, the precision is given by r over sigma square. If it is repeated one time, it is 1 over sigma square, where sigma square is nothing but it is your error variance per unit. Clear? So, then another term frequently used in design of experiment is the efficiency of a design. So, efficiency compares the ability of two designs. For example, if you have two design D1 and D2 and you want to compare their efficiency, compare their ability, which one is more able to detect treatment difference? And variance of the difference between two treatment means, for example, in these two designs, for D1, suppose it is 2 sigma 1 square upon R1, and for D2, it is 2 sigma 2 square upon R2. Then how you will find out the efficiency of D1 relative to D2? So efficiency is a relative terms. So efficiency of design D1 relative to design D2 is just divide the variance of difference of these two. So R1 sigma 1 square divided by R. If you divide this thing, efficiency of D1 relative to D2. Okay. So how you can find out? So you will put this thing in the denominator. So how? So it is twice sigma 1 square divided by R1 over twice sigma 2 square over R2. So if you simplify it, then I think this formula is, this is R2 and this is R1. So this ratio will give you the relative efficiency. And interpretation means if E is equal to 1, both design are equally efficient. If E1 is greater than 1, it means D1 is more efficient. If E is less than 1, it means D1 is less efficient than D2. And how you can increase the efficiency? So you should decrease sigma square in order to increase the efficiency. Simple formula. So decrease sigma square. The sigma square can be decreased if you use homogeneous blocks and reduce experimental error variance. Or another way, you can increase R. If anything is divided by more quantity, this should be less. So increase R, increase the number of replication in order to get more precision. Right? Then another term is factors. So factor are experimental variables under study. These factor may be fertilizers, feed rations, or fungicides. So level of a factor, the levels of a factor represents the way it is varied in the experiment. For example, if you have nitrogen as a factor and phosphorus as a factor, so various levels, say N1, N2, N3, these are the three levels of nitrogen. Means 10 kg, 20 kg, 30 kg. Means your levels are increasing, varied. And similarly, P1, P2, P3. Various doses of phosphorus, say 20 kg, 30 kg, and 50 kg. So these are various levels of a factor. So two levels, if a factor has two levels, drip versus sprinkler irrigation. If three levels, nitrogen application at 0, 60, 120 kg, and so on and so forth. So level of a factor represents the way it varied in an experiment. Then uniformity trials. What are uniformity trials? Uniformity trials are conducted by planting a single crop uniformly across an area and maintaining consistent cultural practices, that is the package of practices. The area is divided into smaller plots and individual yields are recorded. Okay. Now, what are the uses of these fertility trials? The fertility trials are conducted to create yield maps to guide field layout. Number two, these trials determine the shape, size, and orientation of the plot so that efficiency can be increased. Number three, they are used to group the plots into blocks of similar fertility gradient. And number four, they optimize plot size for future experiments. So, before conducting the experiments in a particular land, the fertility trials are conducted in order to create the yield maps of that land. And with the help of these trials, you are able to determine shape, size, and orientation of the plot so that the uniformity can be maintained. You can group with the help of fertilized trial into blocks for similar fertility, and you can also optimize plot size for future experimentation. Then next part, I quickly recap agronomic trials. Agronomy trials study factors like fertilizers, planting time, rate, density, tillage, and pest control. What are the key features of these experiments? They often involves combination of multiple factor levels, various level of nitrogen, various level of phosphorus, and so on and so forth. And they require larger plot due to machinery use and border effect. So in these experiment, the plot size should be large. And some other types of trials are there. There are varietal trials conducted by plant breeding people. The varietal trials are essential for plant breeding programs and they ranges from small plots, means single plot or row, to large replicated yield trial. And blocking is nothing but it is grouping of experimental units into homogeneous blocks to reduce the variability. And how it works? The treatments are randomly assigned within blocks. Number two, if you take an example, if field has two soil types, plots are grouped by soil types and treatment are randomized within each group. Then what is your goal? Goal may be maximize variation between the blocks and minimize the variation within the block. So thank you very much for your patience. So these are the few key component of design of experiment, its terminology, its basic principle, and all these discussed in this lecture should be keep in mind in order to plan your experiment, in order to answer your research questions, in order to formulate your hypothesis, in order to conduct the uniformity trials, the number of replications, and so on and so forth. So I think this video is helpful for you. If you find it helpful, please subscribe my channel so that next videos notification can be sent to you in time so that you can view another video of similar kind. Thank you very much. Thanks a lot.